Good afternoon, everyone. My name is Zijun Wang. I'm a postdoc from Michelle Newson's Wax Lab at Rockefeller University, and very thanks for this great opportunity where I can share some of our recent work on the COVID-19. Today, my topic is B-cell responses to SARS-CoV-2 in the setting of COVID-19 infection and vaccination. According to most updated, SARS-CoV-2 has infected more than 120 million individuals, which results in over 2.65 million deaths worldwide. This all contribute to a big public health issue. Starting from the last March, which is almost the beginning of the pandemic in the US, our lab has been recruiting convalescent individuals who were infected with the SARS-CoV-2 to check both their polyclonal and monoclonal B-cell and antibody responses to SARS-CoV-2 after their infection. Previously, we have characterized the antibody responses at 1.3 months post-infection, and further, we found an evolution of the anti-SARS-CoV-2 antibodies at 6 months post-infection. So recently, we are also looking at the 1-year time point to investigate how amazing our immune system is. In this current study, there are 63 convalescent individuals involved, including 37 who didn't receive the vaccine yet, and 26 who received either the Moderna or the Pfizer mRNA vaccines. The sample collection date of this vaccine is around 40 days after the vaccine. In this cohort, the individuals that returned for a 12-month follow-up, we found 10% had been hospitalized, and the others had relatively mild initial infections. 40% of the individuals has persistent long-term symptoms after 12 months, while the percentage of this we know from 6 months post-infection was 44%. And we found here that the symptom persistence was not associated with severity of acute disease and the duration or with vaccination status. Also, we found that individuals that had not been vaccinated maintained their anti-RBD IgG, IgM, and IgA titers between 6 months to 12 months. And we found a slightly decrease in the IgA titers, while the vaccination increased levels of all three isotype titers, especially in the IgG component. Well, there is an average of 5-fold increase in IgG titers in the vaccinated in participants. And the two exceptional individuals who did not show an increase had been vaccinated only 2 days before sample collection, which are shown here in the purple. While the anti-nuclear capsid antibody titers decreased significantly between 6 months and 12 months, no matter if they were vaccinated or not. So our summary in this part is the persistence of humoral immunity to SARS-CoV-2 viral antigens is different in each individual, and the longevity of anti-RBD antibodies response is better than the anti-nuclear capsid. We then measured the plasma neutralization by using an HIV-1 serotype virus with the SARS-CoV-2 spike protein. 12 months after the infection, we found in our cohort that the geometric mean of the NT50 for the unvaccinated individuals was 75, which was not significantly different from the same individuals at the 6.2 months. However, the vaccinated individuals showed a geometric mean NT50 of around 3,000, which was about 50-fold higher than vaccinated individuals and significantly increased compared to the 6-month titer. We also found that plasma neutralizing activity was directly correlated with anti-RBD IgG titer, which is not correlated with the anti-nuclear capsid titers. Next, we check the plasma neutralizing activity against the circulating variants of concern or interest. We perform neutralization assays on serotype virus of these variants of concern or interest, including the B117, which is the UK variant, and the B1351, which is the South African variant, and also the B1526, which is the New York variant. We found that 12 months after the infection, neutralizing activity against the virus was generally lower than against the wild-type SARS-CoV-2 in the same assay, especially with the South African variant. But what's inspiring is that after vaccination, the NT50 increased 30 to 60 folds against both the wild-type SARS-CoV-2 and the virus. Next, we check the memory B cell compartment. We first measure the RBD binding B cells by flow cytometry by using the RBD as a bait with two different fluorophore at 3 different time points in both non-vaccinated and vaccinated people. We also measure the B cells that bind to the wild-type RBD and also the CAM mutant RBD, which are mutations that presented in many variants of concern. We found that the number of RBD-specific memory B cells was slightly decreased between month 6 and month 12 in the unvaccinated people, but it was increased more than 8 times in vaccinated group. The number of the variants RBD cross-reactive B cells was lower at each time point compared to their binding to the wild-type RBD. But it shows the same tendency over time. Vaccination also significantly increased the cross-reactive B cell compartment. Previously, we have already deciphered that 6 months after the infection, the antibodies from the same individuals are still evolving from 1 month post-infection. Now, in order to know if the memory B cell compartment keep evolving at 12 months time point, we sequenced the same 10 individuals at 12 months post-infection, and of which 4 are non-vaccinated, while 6 are vaccinated. In this pie chart, for each individual, the slides in same color indicate a conserved clone that presented at more than 1 time point, and the gray indicate new clones, and white slices are persistent singulets. These black outliers indicate the percentage of the clonal expansion among all the sequences that were obtained. And so from this pie chart in both non-vaccinated people and vaccinated people, we both see that the antibody evolution after 1 year or so. We also found that all individuals assayed at 12 months showed expanded clones of RBD binding memory B cells that expressed closely related VH and VL genes. So in this circles plot, all the lines in different colors links two antibody sequences, and they use the same IgVH and VL and JH and JL gene. We then checked more closely to the vaccinated individuals. We found an increase in size of memory B cell compartment, which accompanied with an increase in the absolute number of B cells presenting persistent clones. As shown here, we found the number of clonal expanded B cells both in non-vaccinated and vaccinated people. The dots show that the dots show in same color, it's a clone presented at more than 1 time point. And we found that in unvaccinated group, the number of clonal expanded cells are quite similar over time, while there's a significant increase at 12 months in vaccinated individuals. And the antibody variable region genes of both heavy and light chain genes are gaining somatic happy mutations at 12 months after infection, which also indicate that the antibodies evolution over time. So our summary in this part is that clonal re-expansion of memory B cells in response to vaccination is not due to additional accumulation of somatic happy mutation if the clones were re-entering and proliferating in germinal centers. In order to know how these antibodies behave, we cloned and expressed more than 150 recombinant monoclonal antibodies at this time point, 1 year after the infection, from 6 of the individuals. And there are three categories of the antibodies that we selected to express, including the ones that were randomly selected singlets at 12 months time point, and also include the ones that appeared as expanded clones or singlets at more than one time point. And also we select some that were newly arising expanded clones. Among the antibodies that have been tested, 99% of the antibodies bind to the RBD, which indicate that our flow cytometry method used to identify B cells that express anti-RBD antibodies was very efficient. And we found the geometric mean of the EC50 of the antibodies obtained at 12 months was 2.6 nanogram per ml, which was significantly lower than that obtained from 1 month and 6 months antibodies. But there is no difference between the vaccinated and or non-vaccinated. The increase in antibodies binding activity may suggest that an increase of their affinity, which will be further illustrated in the following experiments. We next examine the neutralization activity of the monoclonal antibodies. So in here, each dot represents a monoclonal antibody, and the number in the middle of the pie chart shows how many monoclonal antibodies were tested in each assay. And we found that the geometric mean of the IC50 at 12 months time point was improved from 171 nanogram per milliliter at 1 month time point to 116 nanogram per milliliter at 6 month time point to 79 nanogram per milliliter at 12 months time point. When we further compare the neutralization activity in singlets and in the clones, we found that an increased potency of the 12-month antibodies was especially evident in antibodies from the persistent clones. So the overall increase in the neutralizing activity among conserved clones was due to accumulation of more potent clones and less clones that were not neutralizing. Here, the gray slice indicate the non-neutralizing antibodies. In addition, we found that there was no significant difference in the neutralizing activity of these monoclonal antibodies if they are from the vaccinated or non-vaccinated people. So our summary of this part is that antibodies obtained from clonal expanded B cells after 12 months were more potent than antibodies obtained from unique B cells at the same time point. We then asked whether the loss of non-neutralizing antibodies over time was due to preferential loss of antibodies targeting specific epitopes. We performed the BOI experiments in which a performed antibody RBD complex was exposed to a second monoclonal antibodies targeting one of the three classes of structurally defined epitopes. The classification of the monoclonal antibodies was before shown in Christopher Obami's paper, which published last year in Nature. And we randomly selected 60 antibodies, of which 30 are from the 1.3 months and 30 are from the 12 months. The 60 antibodies were evenly distributed between the two time points and between neutralizers and non-neutralizers. And also these antibodies are with comparable neutralizing activity from the two time points. In order to know how these monoclonal antibodies bind to the SARS-CoV-2, we use the competition BOI to characterize. Here we select C105 as a class 1 antibody, C121 and C144 as class 2 antibody, and C135 as class 3 representative. And in this table, there are 3 different colors showing here. So the red means that the 2 antibodies, they didn't compete at all. And the orange indicate that there are slight or medium competition. And the gray means that these 2 antibodies compete each other and they bind to the same epitope. So here we found the interesting thing is that non-neutralizing antibodies fail to inhibit binding of class 1, 2, or 3 antibodies, and therefore they must bind to epitopes that do not overlap with the epitope of this class of antibodies. And in contrast, Most of the neutralizing antibodies blocked class 1 or 2 antibodies, which targeting epitopes are structural components of the RBD that interact with the AC2. And in addition, we found 9 of the 15 neutralizing antibodies blocked both class 1 and class 2 antibodies, and only 1 of the 15 obtained after 12 months did so. And in contrast to the early time points, 13 of the 15 neutralizing antibodies obtained after 12 months only interacted with C121, which is a class 2 antibodies. We also measured these antibodies' affinity to the RBD, and we found the affinity were similar among neutralizers and non-neutralizers at both 2 time points. While we can see that although the difference was small, both neutralizers and non-neutralizers showed increased affinity at 12 months time point. Next, we asked whether there is an increase in neutralization breadth over time. We assayed the neutralizing activity of these monoclonal antibodies against a panel of different RBD mutants governing the mutations that contained in the circulating variants. And we found that the increased activity was evident against most of them. For example, the K417N, E4A4K, and the N501Y. So our summary of this part is the neutralizing antibodies are retained, and non-neutralizing antibodies, which target the RBD surface that do not interact with the ACE2, are gone from the repertoire over time. And also we found that the evolution of the antibody repertoire results in acquisition of neutralization breadth over time. And we next checked whether the change in specific clones over time are associated with increase in affinity and breadth. We measured the relative affinity of 15 pairs of antibodies from clonal B cells that were maintained over time. So there are 3 categories. So we selected 6 clonal pairs showed equivalent neutralizing activity, and 8 showed increased neutralizing activity, and 1 showed decreased neutralizing activity against wild-type SARS-CoV-2 after 12 months. And we found there was a significant increase in overall affinity over time, including some antibodies with no measurable neutralizing activity. We then checked the neutralizing breadth for the 15 pairs of antibodies, which targeting epitopes that can be classified into 3 dominant classes of neutralizing antibodies. However, neutralizing breadth increased between 1.3 months and 12 months in all 15 pairs, even when neutralizing activity against the wild type was unchanged or decreased. Only 1 of the 15 antibodies obtained after 1.3 months neutralized all the mutants tested. In contrast, 10 of the 15 antibodies obtained from the same clones after 12 months neutralized all variants tested with IC50 as low as 1 nanogram per milliliter against the triple mutant K1 CAM mutant found in South African variants. So our summary here is the persistent clonal evolution of anti-SARS-CoV-2 antibodies contributes to better potency and breadth, thus resulting in monoclonal antibodies with exceptional activity against a broad group of variants. Our conclusion of this study is that immunity in convalescent individuals is long-lasting against SARS-CoV-2 for at least 1 year. And mRNA vaccines boost antibody responses and expand the memory B cell compartment, which are protective against circulating SARS-CoV-2 variants in naturally infected individuals. Thanks for the great help from Rockefeller University, especially Michelle Newton's lab, and our great hospital program to recruit all the patients and all the virus neutralization assays that have done in Paul Binash's lab, and also all the structure that characterized in Caltech in Pamela Belkman's lab. And thank you for everyone for your attention. Thanks.